Hello, everyone. My name is Sairam Behera. I work as a research fellow in the Sedlazeck lab in the Baylor College of Medicine. First of all, I would like to thank LabRoots for giving me the opportunity to talk about my current research on scalable comprehensive genomic analysis. In today's talk, I will be talking more about the small and large variant analysis. So talking about genomic variants or the variant culling, so this is a workflow we use starting from the library preparation from the samples, then we send it for the sequencing, then we get the sequencing reads, then we use the alignment and variant detection tools to get the variants. That is typically in the VCF file format. So during the alignment stage, we align the sequences to the reference genome, and from that we get the BAM file that is used as an input to the variant detection tools. So talking about the reference, we all know that Human Genome Project was completed in 2003, and the GRCS 37 and GRCS 38 references are mostly used for most of the studies. And a few years back, the T2T consortium has released the most complete version of the human reference that is called the CHM13, and it contains almost 200 megabase per more regions, and also that resolves some of the errors in GRCS 38 reference. But a lot of work to be done to use that one because we need the genome annotation and some other resource files. But the good thing is that there are some tools available. For example, this LeviosAm2 that could lift over the T2T mapping coordinates to the GRCS 38 reference. And also, there is another good news is that the Genome Enable team has released a modified version of GRCS 38 reference. That was based on the refinement of our previous work that was published in Genome Biology, is this Fix-It Felix paper. So where it resolves the errors of the GRCS 38 reference by masking the false duplicated regions and also adding the decoy sequences for the falsely collapsed regions. Also, it marks the contaminations as well. So the next question is that if a single reference is enough to study all the diverse populations, and also what if the genome is more different from the reference? In that case, prediction of the genetic risks may not work pretty well. So for that, the Human Pangenome Project has released a genome sequence of 47 people, and that is a collection of the reference from the diverse human populations. And you could see there was some news for this release, and so here is a link for the CNN Health that says the scientists have updated the human genome to make it more inclusive. And this figure basically shows the representation of the pangenome. But the question is, how can we use it? So a lot of work to be done for that, and we need to develop a lot of tools like for the alignment as well as the variant detection tools. So when I talk about genomic variations, we broadly classify them into these four categories. The first one is the small variants, which is typically less than 50 base pair. That includes the point mutations like SNPs and the small insertion and deletions called indels. And the next one is the large variants, usually called structural variants, that's greater than 50 base pair. And that includes these five categories: insertion, deletion, inversion, duplication, translocation. The another type is the copy number variation. So that could be a subtype of structural variant, and it includes the duplication and deletions. And the last one is the short tandem repeats, where the short segments are repeated many times in the reference. So all these variant types are present in a genome, but most of the studies, they often focus on these SNVs, and most of the studies also based on subset of variant types, and they study it independently. But the problem is that if we study them independently, the link or the relation between different variant types is lost. So the question is how to address these challenges. And we know all these variants are important, so there is a need to push the genomics forward by analyzing every variant together. So that's why this comprehensive genomics, where we could perform the joint calling as well as the analysis of all the variant types. But the question is, can we do it? Or also we can use the pan-genome concept. So that is kind of the need of our right now to push the genomics forward. So Illumina's secondary analysis platform called DRAGEN. So that is one of the framework that can perform such tasks. So that implements also the ideas of pan-genome. And the good thing is that it takes only 30 minutes for a 30x whole genome sequencing to generate all type of variants. So I'll be talking mostly about the version 4.2 of DRAGEN. So that includes many components. For example, the SNP, the structural variant, the copy number, and STR, the short tandem repeat colors. Also, it includes 9 targeted gene colors. These genes are highly complex and difficult to map regions. And also for the population cohort study, it includes a GVCF genotyper that performs the joint calling. And it uses the sophisticated hardware to accelerate the alignment and variant culling, which are some of the time-consuming steps in this process. And it offers the complete end-to-end solution where we can give the input in form of FASTQ, BAM, or CRAM, and it can output the VCF and the JSON for all colors. So first, we wanted to see how it performs for the small variant calls. So what we did, we took the NIST Genomics benchmark set, which is considered to be the truth set for the SNVs for this SG002 sample. Then we compared against two different variant color tools, the GATK as well as the deep variant. And for the deep variant, we used both BWMM as well as the GIRAF, which is a pan-genome-based aligner. And if you see this figure for both for the SNPs and indels, the DRAGEN performs a little better than all other tools. And if you look into the F scores, most of the tools, their F score is more than 99%. But if you look into the raw counts, the false positive and false negatives, which are in the red, they are less as compared to other tools. The second thing we wanted to do to see the performance of DRAGEN in the challenging medical regions. And these challenging medical regions contains 273 genes, and they mostly contain the SEC-DOP regions, and also 12 genes in these areas are affected by GRCS38 errors. And you could see the right figure here, both for the SNPs and indels, DRAGEN performance was much better than the second best is the deep variant with the GIRAF. And also, we could identify 5 true variants only by DRAGEN, and that was not identifiable by other colors. So then we wanted to check whether this improved performance only SG002 specific. So we wanted to use other samples as well. And we took the benchmark set for SG001 to 7 from Genomics bottle. And then we compared against other tools. You could see the right corner shows the recall precision and F measure for all tools, all samples taken together. And you could see DRAGEN 4.2 performs better than the other tools. So one of the key things that was used in DRAGEN is the multi-genome graph alignment, which improves the alignment. So it basically takes 64 population haplotypes, also the GRCS38 modified reference genome, and it maps to the primary reference, for example, here in this case about GRCS38, and also the secondary, the context, those are the haplotypes. And based on both the mappings, it adjusts the alignment, it adjusts the mapping quality as well as the mapping score. And the good thing is that it takes only 8 minutes on this on-site DRAGEN server. Basically, the alignments are always the time-consuming steps. So the next thing we wanted to see if it also works well for the structural variants. For the structural variants, we compared DRAGEN against these 3 other tools, MANTA, DELI, and Lumpy. And you could see for the genome-wide, the insertion variants, DRAGEN has better performance than MANTA and DELI. Lumpy didn't report any insertion. And MANTA is also developed by Illumina. And we also did the same for the CMRG regions. Both for the CMRG regions and the genome-wide, we also see for the deletions, which is little easier than the insertion identification. DRAGEN also performs better than all other tools. And this is basically showcasing DRAGEN's ability to detect SV gene also in repetitive regions or the complex regions. The next thing we did for the copy number variations. For the copy number variations, we have either amplifications or deletions. So in the amplification, we have more copies as compared to the reference. And for the deletions, we have less number of copies as compared to reference. And for the copy number variations, we don't have any benchmark set, but what we did, we collected all the deletion variants greater than 1 kilobase pair from the Genomics truth set for the SVs. Then we compared DRAGEN against the CNVinator, another tool for calling the copy numbers. And you could see for the 1 kilobase to 5 kilobase regions, DRAGEN performs much better than the CNVinator. So this is because DRAGEN uses the discordant and split read signals from the SV calling. This is how you can connect different type of variants. And also it utilizes a modified shifting level models. And for the 10 kilobase pair until greater than 50 kilobase pair, both CNVinator DRAGEN performs almost similar. But that's the better performance in the 1 kilobase to 10 kilobase regions. The other thing what we did, benchmarking the short tandem repeats. And short tandem repeats are very useful. Here you can see an example where the expansion in the short tandem repeats that causes the Huntington disease. So for the short tandem repeats, DRAGEN uses expansion hunter tool that was published in 2019, and it uses a predefined repeat catalogs. So you can see the repeat catalog in this figure, the first one. So that basically is collected from the literature, and you can see there is a gene and the repeat units and the regions where these repeats can occur. So DRAGEN uses this repeat catalog to align the reads to these regions, then report the STRs. And for this one, for the analysis, we use this 174K region repeat catalogs. So what we did, we benchmarked these STR calls against recently published genome in a bottle benchmark set for the STRs. So this paper was published only a week ago. And this contains almost 1.7 million repeat catalogs. The benchmark set contains 1.7 million catalogs, but the DRAGEN includes only 174K, and the GANG STR, another tool to call the STRs, that contains 832,000 regions. And you could see the precision is good for the DRAGEN, and the orange one shows the precision for the GANG STR. But for the recall, it does not, recall does not shows good here. That's because there are a lot of regions that is outside of this 174 or 832K regions for both for the DRAGEN and STR respectively. And the middle line shows the DRAGEN call set for the indels. So that is the genome wide, that does not restricted to this 174K region. So for the middle one, the DRAGEN indel calls, the recall is much better than other DRAGEN STR calls. So the next thing what we did, we just wanted to include only the regions that was called by DRAGEN and the GANG STR. So the calls outside the individual catalog regions are not penalized. Then you can see in this the right side red box, the recall and F measure, that improved when we used only the regions for the validations. So that's how it shows, DRAGEN has a better performance for the STR calling as well. So this is where we are showing all the calls from the DRAGEN for this HG002 sample. So this figure shows the distribution of the SNPs and indels. The top this peak, the peak basically shows almost 4 million SNPs in HG002 sample, and the right side and left side shows the small insertion and deletions. This is for the small variants. Then when we included the insertion and duplication, you could see there is a nice allele repeats in the 300 base pair regions, and also you could see there are some large insertion and deletions on the right box. Similarly, we also included the deletions. So you could see again the nice allele repeats in the 300 base pair region on the left, and we have more number of deletions in 1000 base pair to 10,000 base pair. Also, we have some large deletions as well. So this basically gives a comprehensive view of all the variants by taking their length into considerations. We did the same for all other samples. HG001 sample, we saw the similar pattern. So that's similar to the HG002. So the next thing we wanted to see how we can do the population study as well. And when we talk about population study, it could include 1,000 genomes to millions of genomes. So their scalability comes into picture. So for that, DRAGEN provides this GVCF genotyper, which can efficiently aggregate the genotype VCFs to perform the joint calling and genotyping. So this joint genotyping is useful for the GWAS, the rare variant, as well as the phasing and imputation studies. And the good thing is that this the multi-sample VCF file that is generated using this GVCF genotyper contains the cohort level variant statistics like allele frequency, genotype, as well as the genotype quality. And for 3,202 samples, it took around 2 hours on Illumina's phase 4 server with 200 parallel jobs, so which is pretty good. So next we did analyze the 1,000 genome project samples using the DRAGEN, and this 1,000 genome project sample is a widely studied control samples, and it contains the samples from various ethnicities. The first thing what we did the SNV analysis, taking the DRAGEN call for all these 3,202 samples. On the left figure, you can see a PCA plot that shows a nice cluster of variants for all the populations. The orange one is from the American, so that's the admixed population. That's why it is more scattered than the other populations. And the right two figures are basically the count of the SNPs and indels for various populations. And you could see for the African population, both SNPs and indels are much, much higher than the other population. And there are some outliers in the African which are same as the other population as well. The next thing what we did, we just wanted to compare the DRAGEN call set with recently published GATK call set that was published in 2002 in the Cell. And we divided all these the variant calls into three categories: the singleton, where the variants found in once in population, and then the rare ones were the low frequency variants, and the common one, which is the high frequency variants. And also we compare them against the DB-SNP database to know whether they are known or novel variants. For SNPs, you could see both for the DRAGEN, which is in blue, and the GATK is in orange, they are performing similar. That means they are reporting same number of variants for both for the singleton, rare, as well as the common. But we saw a significant improvement of the novel as well as known variants for the indels. You could see on the right figure, the DRAGEN has more number of singleton and the low frequency variant as compared to GATK. The next, we did the comparison of all the variants which has the functional consequence. So this figure basically shows the similar number of variants reported for all different type of functional variants. Next thing, what we wanted to do to study all the large variants for these populations. For that, we use the DRAGEN call for the STR, the short tandem repeats, as well as structural variants, as well as the copy numbers. And the first thing what we wanted to do the unification of all these large variants across 3,202 samples. This is needed because the variant types are not disjoint, and DRAGEN produces separate VCF files for the STR, SV, and CNVs. So we use the Truhari tool to reduce the redundant variants and then merge them at the sample level. And then after merging all different type of variants at sample level, we use the BCF tool merge to merge all samples together. But one thing we wanted to check to see if we are losing any information by doing this merging, so whether we are doing the over merging or under merging. So for that, we evaluated the SG002 sample against the genome in a bottle truth set. And on the right side figure, you could see dark blue and light blue that is showing the before and after merging. And we saw the precision Likert and F scores, they are almost same before and after merging. So that basically gives us confidence that we are not losing much information by doing this merging. So after that, we did the analysis for all the populations taken together, and you could see on the left side is the PCA plot that shows the clear distribution based on the population. Again, orange has the scattered points that shows the admixed population in American population. And similar to the SNPs and indels, you could see for the African population has always a higher insertion and deletions. So the next thing we wanted to check the targeted colors in the DRAGEN. So as I mentioned earlier, DRAGEN has 9 targeted colors, and they are very complex regions and difficult to map regions. And DRAGEN utilizes a common SNV in population to estimate the copy numbers, and it also reports the recombinant events if they don't follow the general phasing pattern. The benchmarking of all these colors was done using the orthogonal dataset as well as the long read based calls. And the HLA typing for the HLA color was also checked against the HiSAT, T1K, and HLA-ASM tools, and they were highly concordant. And the last one, this LPA, that was pretty interesting. We have been also studying the LPA for some time using the DRAGEN LPA color. So let me talk a little about the LPA. This LPA gene is very important because the particle called LP, the lipoprotein, is encoded by the LPA gene. And if you look into the figure here, that shows the different regions, KIV regions of the LPA. And the most important is the KIV2 regions, which is in orange. So this KIV2 region has repeats, and that is important to the lipoprotein isoform size. The high number of the repeats in this KIV2 region is associated with low lipoprotein concentration. And this low lipoprotein is associated with cardiovascular risk. And you could see there is a Washington Post article about this lipoprotein and why it is important. And this is a region in the chromosome 6. And again, as I mentioned, it is a very complex region and difficult to map. And you could see here the IGV screenshot of the LP regions. And this is a mapping from all different sequencing technologies. And the middle one is the Illumina, the short reads, where you could see a lot of blank spaces that shows the low mapping quality. Similar performance also we saw for the PacBio Hi-Fi as well as ONT. But you could see the coverage is so high because in the GRCS 38 reference, it has only six copies. And all these repeats are 5.5 kilobase pair long, and it can occur 5 to 50 plus times. And there is a dedicated tool for LPA in DRAGEN, and this is, to the best of our knowledge, this is the first time we have a dedicated tool for this LPA. And we benchmark the DRAGEN LPA color against the BioNano, and the good thing is that the DRAGEN can also report the allele-specific calls for 50% samples, which is very, very, very important for the further analysis. And what we did an association study of this KIV2 copy numbers against this lipoprotein concentration. And you could see on the left figure that was done for the African American, European American, and Hispanic American population. And we took 1,000 samples each, and these all are coming from the cardio risk patients. And you could see for the low LPA concentration, we always see the high copy numbers. The mean LPA for the low copy numbers, we could see a high mean LPA levels, and that is consistent in all these populations. But for the European American and Hispanic American, we saw a steep decline. You could see on the figure A, and same thing also we did by dividing the LPA concentration into three clinically thresholds, like low, medium, and high. Low is the one where the LPA level is less than 75 nmol per liter, and high is the one which is greater than 125 nmol per liter. And the right figure you could see for the low lipoprotein level, we have a high KIV2 copy numbers. And that is also consistent across all the Hispanics. So that basically confirms whatever we have in the literature studies, the association between the copy number and this LPA concentration. The next thing what we did, we also did the association against two allele-specific copy number. As I mentioned earlier, DRAGEN was able to report 50% of the samples with this allele-specific copy numbers. And this figure is quite busy, but you could see all these dots represents the low, medium, or high the lipoprotein concentration, and the y-axis is allele 2 copy number, and x-axis is allele 1 copy number. And allele 1 copy number is always taken, which is the highest among allele 1 and allele 2. And you could see this allele 1 copy number which always outweighs the other. So you could see on the European American, this little the right box where the allele 1 copy number shows associated with low LPA concentration, where the allele 2 is pretty low, which is very interesting results. So this basically shows why the allele specific copy number is also important in some of the association studies for the LPA. So all these results are currently in the preprint right now. So we submitted the LPA paper to BMC Genome Medicine, and the DRAGEN analysis was in revision in Nature Biotechnology right now. So this contains all the details analysis of whatever we did for the LPA as well as the DRAGEN analysis. So to summarize my talk, we showed that a scalable comprehensive analysis, including different type of variants, is very, very useful. And this is also way to push our genomics forward. And DRAGEN performs better compared to other state-of-the-art short read-based colors, both for the SNVs, the SVs, STRs, as well as CNVs. And the targeted colors in DRAGEN will provide further insights into some complex clinically relevant genes. And we showed that LPA color estimates the CNVs accurately, and also it gives the allele-specific copy number for almost 50% of the samples. And the association study we did with the lipoprotein level and the KIV2 copy number, and that also showed a strong correlation between this LPA concentration and the copy numbers. In future, we are working on the somatic and mosaic colors with DRAGEN's next version, which is DRAGEN 4.3. And also we are doing some analysis on 30K cardio patients using this DRAGEN call sets. With that, I would like to thank all my team members in Sedlazeck lab, as well as all the people in the Human Genome Sequencing Center at Baylor College of Medicine. And also we collaborated with some people from UT Health, and also the Illumina teams. Of course, they have been collaborating with us for some time. And with that, I would be happy to take some questions. Thank you.